For folks who are joining, we'll be starting in a couple minutes. We just want to give time for people to get through the waiting room and to log on. For folks who are joining, we'll give it just about another minute and then we'll get started. So thank you for being here today. We'll get started in a minute. Just there's still a few people joining, so I'm going to give them a chance to participate fully. Good morning, good afternoon, good evening, everyone. My name is Ken Mayer. I'm part of the Harvard Center for AIDS Research and also work at Fenway Health. And with my co-moderator, Dr. Patrick Sullivan of Emory University, CFAR, we're delighted to invite you to this ART for Prevention webinar series. Before introducing our speaker today, I just want to make sure people have on their calendars August 4th, noon East Coast time. We'll be having a talk by Mitchell Warren, who is the founder and the head of AVAC, and we'll be talking about sort of community responses to all the disruptive changes that have been going on for the conduct of our research and the delivery of services related to HIV prevention activities. So Mitchell Warren, August 4th. So it's really a pleasure to introduce Dr. Urvi Parekh of University of Pittsburgh, where she is an associate professor of medicine and has led a number of NIH and other federal government and foundation supported efforts to understand the evolution of drug resistance that we may be seeing, particularly in individuals who acquire HIV on PrEP, but other landmark work in this space. So we're very happy to have Urvi with us today. We will have time for questions and answers at the end of Dr. Parekh's talk, but if you do have questions, you should feel free to put them in the chat. Patrick Sullivan will be monitoring that, and if you have a burning question or clarifying question, you can hit the raise hand function. We'll try to address that as well. But Urvi, we look forward to hearing about some of your initiatives, including the Mosaic project and some of the other exciting work you've been doing. So thank you.
Thank you so much. Thank you to the Harvard CFAR working group for the invitation to speak with you today. I'm pleased to be here, and I'll be talking to you today about monitoring drug resistance and treatment outcomes in individuals who acquire HIV on PrEP and routine care. So especially in the current times, it's good to remember that HIV prevention remains a key pillar for addressing the HIV epidemic, both domestically and globally. We know that PrEP is highly successful at protecting adherent individuals from acquiring HIV, although we have seen breakthrough HIV acquisitions on all PrEP modalities, though this is rare. So in our research, a key focus of our area is to try to understand what is the risk of resistance and impact of future treatment among persons who acquire HIV despite using PrEP. Despite remarkable progress in HIV testing and treatment, new HIV infections continue to exceed US and global targets. With 1.3 million new infections globally in 2023, we're currently not on track to meet the UNAIDS target of reducing new infections to no more than 250,000 in 2025. A number of modeling studies have predicted that the scale-up of PrEP could further reduce HIV incidence. And you can see from this UNAIDS data that the continued need for HIV prevention is still there in addition to expanding access to treatment. And now we are really in an exciting era of expanding PrEP products. I've highlighted here 3 that are approved for use in the US, 1 that is in FDA review, and 1 that is approved globally, but not in the US. So just to orient us to all the products, this includes oral daily tenofovir disoproxil fumarate with emtricitabine and oral daily emtricitabine with tenofovir alafenamide. The latter is for people assigned male at birth. We have long-acting injectable cabotegravir, which is given as an initial injection followed by a second injection at 4 weeks, then bi-monthly afterwards. The newest one expected to be reviewed in a couple of weeks is injectable long-acting lenacapavir, which is a twice-yearly injection. And I won't be covering dapivirine ring today. It's not FDA approved in the US, but it's a monthly vaginal ring available in select countries in Africa. A key challenge with PrEP is that the same drugs and drug classes are used for both HIV prevention and treatment. So for example, for HIV prevention, we use TDF, FTC, and FTAF, which are a key component of NRTI-based antiretroviral therapy. Cabotegravir is used as cabrilpivirine in the US, and other INSTIs, including dolutegravir, is used as first-line ART in many parts of the world as TLD, or dolutegravir lamivudine tenofovir disoproxil fumarate, or another INSTI, bictegravir, is used with FTAF. And lenacapavir, which is a capsid inhibitor, is also used both as prevention and treatment. So this can be a concern if drug resistance from PrEP leads to virologic non-suppression on ART and secondary transmissions. Conversely, transmitted resistance from viremic individuals on ART could lead to breakthrough HIV acquisition on PrEP and secondary transmissions. So resistance with HIV acquisition on PrEP could occur through 3 major pathways. One is if someone starts PrEP during undetected acute infection. Most diagnostics test for HIV and detect antibodies, so this could occur if a person is infected but has not seroconverted yet. A second is if they continue using PrEP after breakthrough infection before HIV is detected. HIV acquisition could occur due to insufficient adherence, and there have been rare cases of breakthrough infection despite sufficient drug levels. And finally, PrEP could fail to protect against a transmitted resistant strain from a partner, although currently this would be rare for cabotegravir and lenacapavir, since circulating resistance in those 2 drug classes are negligible. So in today's talk, I want to address a few key questions. One is a question about oral PrEP. This is the most widely used PrEP in countries in Africa, and so we'll dive into the question of whether, or among individuals who acquire HIV on oral PrEP in countries in Africa, what proportion of PrEP-associated resistance is observed. And this is important because first-line TLD contains both tenofovir disoproxil fumarate, which is a component of PrEP, and FTC, or emtricitabine, which is similar to 3TC used in first-line ART. And the second key question to address today is the question, what are treatment outcomes in individuals who acquire HIV on CAB-LA PrEP? We know that INSTI resistance is frequently selected in the rare individuals who do acquire HIV, and how will that impact current treatment? and overall understanding causes of breakthrough HIV acquisition on PrEP and subsequent treatment outcomes can inform PrEP programs for the greater goal of preserving ARVs for both treatment and prevention. So let's start with oral PrEP, which has now been used for over a decade in the US for HIV prevention, and also used globally for HIV prevention. The effectiveness of oral PrEP in preventing HIV infection was established through a number of pivotal placebo-controlled trials. And you can see here, these include Partners PrEP, TDF2, IPREX, FemPrep, Voice, among others. And the take-home message here is that higher oral PrEP adherence resulted in higher effectiveness of HIV protection. It's notable that as PrEP adherence increases, HIV protection effectiveness also increases. Globally, there have been almost 8 million oral PrEP initiations, according to data on PrEPWatch, as of the third quarter of 2024, with many countries at that time increasing and improving PrEP access. Although I would like to acknowledge that these numbers are from the end of last year, and there may be, or likely, there are recent changes in PrEP access that are not reflected in this data. The World Health Organization recommended a cross-sectional, time-limited, passive surveillance approach of all people who have currently or recently taken PrEP with a scale-up of a new PrEP product. We work together with the WHO to think about the best way to do this. They have also highlighted the importance of monitoring for HIV drug resistance with the rollout of PrEP and national programs. And so since 2017, we have been monitoring drug resistance with PrEP through several approaches. One is our main approach, which was to implement a national research protocol to access HIV drug resistance in persons who acquired HIV on PrEP. So we established these national protocols in Kenya, Zimbabwe, Eswatini, and Zambia, and were in the process of establishing these in Lesotho and Uganda as well. A second approach is the third one listed here. We partnered with existing PrEP demonstration projects and implementation projects to add HIV drug resistance monitoring to their protocol or procedures. And then we were also involved in a large implementation study with over 11,000 participants called Catalyst. The objectives of Catalyst were to assess enhanced service delivery, product effective use, and also the rates of HIV infection and drug resistance. We had collected samples from all CAB PrEP users to be able to assess pre-seroconversion samples for timing of infection and resistance development, similar to the HPTN studies. This project was funded by USAID PEPFAR, so unfortunately has been terminated as of January 2025. At the time of termination, we had plans to monitor, we had plans to amend each of the protocol to include 6 and 12-month follow-up visits. So that was in the works to assess treatment outcome, especially in those on CAB LA PrEP, which had just started rolling out in Zambia and Zimbabwe. So unfortunately, I won't have treatment outcome data to share. We were still able to evaluate our data on resistance with HIV acquisition on oral PrEP to date. So we'll talk about that. And before I go into that data, I just wanted to highlight a little bit of the approach we took to set all of this up. This one slide does not really adequately cover the process of all of the relationships we had built and the rapport we had established through all of these years. And I guess I just want to take a moment to really appreciate all of the people whose time and efforts, particularly our country teams, put into making this happen. So really in a brief synopsis, our approach was to first do stakeholder engagement. We met with all of the MOHs in person in each of the countries, joined their relevant technical working groups. We did it at first until we found our country partners who then really led this work. And we also conducted stakeholder buy-in, including meeting with all of the local NGOs, non-government organizations working in the area of HIV prevention, in-country US government groups, and USAID mission. We then, once we had the buy-in from everyone, went into protocol development. And that's where we really created country teams, including a country coordinator, laboratory specialists. We identified a protocol chair from either the implementing partner organization or someone from the Ministry of Health, and formed a protocol team, including technical working group members, and put the protocol approvals through local IRB. So what was really different about this approach was these were locally led protocols. Our role was more for technical support, but the protocol chairs and everyone on the teams were really locally conducting these. So it wasn't like through a network or anything like that. And then finally, we did protocol implementation. We procured and distributed sample collection kits. So in the photo here is an example of what we would call our HIV DR kit, where we provided all the materials and distributed them, sometimes through existing county networks for distribution of medicines. We created resources, manuals, and job aids, conducted training for service providers, as these were all from routine care and not trial clinics. And we arranged for transportation of blood samples to central labs and established local laboratory testing. Pictured here also is one of our amazing country coordinators, Everline Bosek, who's conducting some trainings and meeting with some of the local staff in their clinics. I have here data from the observational cross-sectional study from 2017 to 2025 from 4 countries, Kenya, Zimbabwe, South Africa, and Eswatini. This manuscript will be out very soon, hopefully sometime this summer in JIAS. The protocol enrolled consenting current PrEP users that were defined as having collected an initial supply or resupply of PrEP, and also having acquired HIV per national HIV testing algorithms after PrEP initiation. So this predominantly was 3rd generation rapid tests that were used to diagnose individuals. A blood sample was collected through whatever clinic they had attended, and they used the kit that we provided to collect that, conducted all of the consents, and the sample was sent for resistance and PK tests, as well as a brief demographics and adherence self-report was administered. We tried to do as much testing as possible in country. The PK had to be shipped and was done in South Africa. So who acquired oral PrEP? We had 283 seroconversions reported, and 90% of those agreed to be in the study and enrolled into GEMS Mosaic. Just as a clarification, the study was called GEMS in the early years, and then the name changed to Mosaic, but it all describes the same study. So of these individuals who acquired HIV, 76% were female, 51% were youth aged 15 to 24 years, and 25% self-identified as being part of a known serodifferent couple. So here are the results of this. So of 245 samples that were available, of those enrolled, 154 or 63% were successfully tested. The reasons for no result were that 2 failed to amplify, and 89 had HIV RNA under 1,000 copies per mil, which was too low to sequence at the local or the national labs in each country that we were using. And these results for the resistance that we observed. Half of the individuals had no major mutations, so that was 54%. 10% had PrEP-associated mutations only, and these were defined as having either K65R, which is tenofovir-associated, K70E, that was tenofovir-associated, although we really did not see instances of K70E, and M184-IRV, which is an emtricitabine or lamivudine-associated, or XTC. An additional 12% had PrEP-associated mutations that also included NNRTI mutations, and 24% had NNRTI mutations only. So to put this into context with what has been also seen in other studies, in the phase 3 clinical trials overall, and this would be a compilation of the many trials that had been done that were placebo-controlled, 5% of individuals who acquired HIV during a follow-up visit had PrEP-associated resistance. By contrast, 65% had been retrospectively determined, who had been retrospectively determined to be infected at enrollment had PrEP-associated resistance. So this was a huge difference in rates that really highlighted that if someone starts PrEP in acute HIV infection, the risk of resistance is very high. In a recent study published by Rafi Landowitz and colleagues, they did a pooled analysis of 72 global post-implementation studies and demonstration projects and found that 28% of individuals who acquired HIV during a follow-up visit had PrEP-associated resistance. And there were no samples collected at enrollment, since these are not studies or traditional clinical trial-like studies. So how many were retrospectively determined to be infected at enrollment could not be calculated. And this aligns similarly to what we observed in GEMS and Mosaic, which was 22% of individuals with PrEP-associated resistance. So to summarize the oral PrEP data and resistance with HIV acquisition on oral PrEP in select African countries, overall, the number of seroconversions, 284 reported, remains very low compared to the estimated number of people initiating PrEP. I know the map I showed, it was in the millions. In the catchment area, we estimated that there were greater than 104,000 people who had initiated PrEP. The proportion of individuals who acquired HIV on TDF-XTC PrEP in routine care and have resistance may be higher than what was observed in clinical trials. And this is to be expected since some resistance may be transmitted from a partner. We did see high rates of NNRTI resistance, both NNRTI resistance alone and with TDF-FTC associated resistance. So we know that any NNRTI resistance would have to be transmitted from a partner. Or we suspect that any resistance would be transmitted from a partner and not because that individual themselves had used NNRTIs, although that's a possibility. A second thought would be that we saw that 1/3 of seroconversions occurred in the first 3 months of starting PrEP. Therefore, some people may be starting PrEP during undiagnosed acute HIV infection that could not be ruled out because the use of antibody-only tests may have limited sensitivity to diagnose acute HIV in this setting, and there are no stored samples to go back to try to see if HIV RNA could be detected earlier than seroconversion. So there is only a limited amount of data on ART treatment outcomes in oral PrEP users with resistance who have been exposed to PrEP. The Nadia study, which many of you may be familiar with, demonstrated high rates of suppression with tenofovir, lamivudine, and dolutegravir, or TLD, in individuals with mutations to TDF or FTC. And we do have an ongoing study in Kenya called ACTION, where we have a small number of individuals, at the moment, 10, who have acquired HIV after TDF or XTC PrEP use. Of those, we saw that 9 achieved viral suppression defined as under 200 copies per mil on dolutegravir-based first-line ART by 6 months post-diagnosis. And that included participants with M184V, IRV. And of the 1 person who did not achieve virologic non-suppression, they self-reported low adherence to ART. In the US, many alternate treatment options are available, making the possibility of tenofovir or FTC resistance there's still many options that could be given with people with that resistance profile. So now switching to a discussion of long-acting cabotegravir. Cab PrEP was FDA approved in 2021 for HIV prevention, and this discussion will mostly focus in the US. So I do also want to pause here and acknowledge Dr. Catherine Koss from UCSF, with whom I am MPI on the Sero PrEP project, and I'll be sharing some of the data from that work. So to introduce long-acting injectables for HIV prevention, this prevention modality, including CAB-LA and lenacapavir, are very highly effective and will be key to achieving global targets to reduce HIV incidence. In CAB-LA prevention trials, incident HIV infections were extremely rare. And infections that did occur within 6 months of the last CAB injection were often associated with integrase strand transfer inhibitor or INSTI resistance mutations. And as I had mentioned earlier, INSTIs are used for both PrEP and ART. And the concern there is that resistance selected by CAB-LA PrEP could compromise viral suppression on first-line ART that includes drugs from the INSTI class. So what we know so far about CAB infection comes from 2 phase 3 placebo-controlled trials, HPTN 083 and HPTN 084. Both studies showed a significant reduction in the risk of HIV infection compared to oral PrEP, which led to the FDA approval of CAB as PrEP in 2021. In HPTN 083, there was a 66% reduction, and in HPTN 084, there was a 91% reduction in risk compared to oral PrEP. This table summarizes the infections seen in HPTN 083 by different categories of when that HIV acquisition occurred. So just to highlight the row in blue, that was the category of individuals, category D, who were infected despite on-time injections. And there were 6 individuals in that category, and all 6 of them had drug resistance with major INSTI mutations identified as shown in bold. Overall, looking at all of the categories, there was a 63% risk of resistance of 12 individuals out of 16 having 1 or more major INSTI mutations. And so in the US, as of now, this graph summarizes individuals on CAB, which is still quite low, but was rising in recent times. So in this data that's from Real World Longitudinal Prescription Database, which includes greater than 94% of US retail pharmacies and more than 75% of mail order pharmacies in a recent publication by Mann and colleagues. The red is shown where CAB CAB began to be rolled out as PrEP in the United States, and there were 13,709 prescriptions as of 2024. So that's just, again, putting it in context with the next slides where I'll describe a few cases of HIV acquisition on CAB. So SeroPrep was a study of HIV infections on PrEP to better understand causes of HIV infection among persons using PrEP and evaluate their treatment outcomes at 6 and 12 months. We conducted this study to help inform future guidance. The study is led by Dr. Catherine Koss at UCSF and myself, and is an ongoing national study, observational, that's enrolling persons across the US with HIV acquisition despite the use of oral or injectable PrEP. We enroll individuals with confirmed or suspected HIV infection while using any form of PrEP. We can enroll participants from any site providing PrEP across the US, and we receive referrals from CDC PrEP line, PrEP clinics, and health systems. As part of the study, we collect blood samples for sensitive diagnostics, resistance, and PK assays. In my presentation today, I'm mostly going to focus on the resistance aspect. Once enrolled, we follow participants to assess provider-selected ART regimens and treatment outcomes in routine clinical care. Our research tests are conducted at the University of Pittsburgh and University of California, San Francisco, and include sensitive assays like single copy assay for quantification of low-level RNA, single genome sequencing of full-length integrase for sensitive detection of INSTI mutations present at 1% or higher frequency, and liquid chromatography tandem mass spectrometry to quantify CAB levels in plasma. As mentioned, today I'm going to focus on the resistance results. And in the data I'll present, we have a few key questions that we are addressing. One is whether INSTI mutations are selected with HIV acquisition on CAB-LA PrEP in routine care in the US. And second, if individuals who acquire HIV on CAB-LA PrEP achieve viral suppression on their treatment regimen. So I will present some of our early data from SeroPrep on a subset of participants. We have a lot more enrolled, but I'll focus on 7 participants on which we have diagnostic resistance and ART outcome data that's fully available. So just to give a little bit of background on the resistance evaluation. There are 2 considerations with INSTI resistance. First, INSTI resistance can occur through multiple pathways, resulting in different combinations of mutations. The 2 categories of INSTI mutations include accessory mutations. These have little impact on resistance unless present in combination with major mutations. And while major or signature INSTI mutations can cause higher level resistance, especially with the accumulation of 3 or more in combination. So for example, Q148R is a common emergent mutation in CAB rilpivirine ART or CAB PrEP trials, and it's observed in individuals without viral suppression or in breakthrough infection. The accumulation of multiple INSTI mutations can lead to higher level resistance. Second, the frequency of INSTI mutations may be important. High frequency mutations are those detected at 20% or higher. typically by standard Sanger sequencing using commercial genotyping assays, while low-frequency mutations under 20% are detected by some commercial NGS assays or by research assays like single genome sequencing. The clinical impact of low-frequency INSTI mutations observed with HIV infection on CAB PrEP is an ongoing area of investigation. So in the next few slides, I will present 7 cases of individuals who acquired HIV on CAB PrEP. And in this first table, these were individuals who were suppressed on darunavir-based ART. So in this table, you see 4 participants. In the second column is their diagnostic result, either antibody antigen laboratory-based assay or a point of care assay, such as a rapid. And in the next column are the major INSTI resistance associated mutations as detected either by commercial genotype or single genome sequencing. So what you see here is that all 4 of these participants had negative serology tests, but were identified as HIV infected based on their HIV RNA that was done with each injection. Their commercial genotype, 3 showed no mutations, while 1 had 3 major INSTI mutations detected. But doing more sensitive testing, 1 of the 3 had additional INSTI mutations detected at very low level. Only 1 genome out of 56 sequenced had E185K, and the second participant seen at day 7. This mutation was not seen again at day 13, N155 and K mixture was seen only in 1 out of 43, and at day 166, G140R was seen in 1 out of 41. So looking at their outcomes, the first column in blue shows the number of days between first detected RNA to ART start. Most individuals started within 2 weeks of their first detected RNA. This 4th individual had a delay of 56 days, which may have increased that person's risk of INSTI mutations. And then the last column is the most recent RNA after how many weeks on their current regimen. So all of these individuals started darunavir, cobicistat, FTAF, and remain on that regimen, and all achieve virologic suppression under 200 copies per mil with a range of 8 to 48 weeks of follow-up. The next 3 participants I'll describe all had low frequency major INSTI resistance associated mutations, but all of them maintained viral suppression after switching to an INSTI-based ART in routine care. Participants 5 and 6 were diagnosed after on-time injections, and participant 7 was diagnosed during the tail phase after that person had already discontinued CAB. Excuse me. All of these 3 participants did have serology detected as reactive antigen antibody, and they also had HIV RNA as detectable. And a commercial genotype of the 2 out of 3 that had results available, no mutations were observed, but all 3 had low frequency mutations observed using single genome sequencing. The first individual had 12 of 40 sequences with Q148R and 1 of 40 with R263L at day 12. These were no longer observed at day 55, where G118R was observed in 2 of 15. Q148R was observed in 2 of 22 sequences at day 2 in participant 6, and S147G was in 1 to 45 in participant 7. These individuals all started darunavir, cobicistat, FTAF, and then switched, eventually landing at the regimen described here. So all of these individuals started ART within 2 weeks of HIV diagnosis. And as of their most recent viral load performed, all of them had suppressed less than 200 copies per mil. The first individual had been switched to Victegravir FTAF and had been on it for 31 weeks and has suppressed HIV RNA. These very low copies were detected by single copy assay, which is a research assay and not by commercial RNA. This individual, these next two individuals, 6 and 7, switched to cabrolpivirine. Participant 6 has been on it for 13 weeks. Participant 7 has been on it for 20 weeks, and both are suppressed on cabrolpivirine. So overall, among a subset of participants diagnosed with HIV during CAB-LA PrEP use in routine clinical care in the US, who subsequently enrolled in the SeroPrEP study, so these are 7 participants that I showed you, 4 of 7 had non-reactive HIV antibody antigen at first detectable HIV RNA. 6 of 7 with INSTI-associated resistance, they had resistance detected by standard sequencing, 4 by SGS, and 2 had no mutations, but all suppressed less than 200 copies per mil on ART, 4 on darunavir containing ART that stayed on that regimen, and 3 who had switched to an INSTI containing regimen. So to put this in context with other data, this table shows data presented recently at CROI by Rafi Landowitz describing treatment outcomes in HPTN 083 participants. The data includes participants from randomized blinded phase 3 studies, the first year after unblinding, and the open label extension. And here, 47 participants that were diagnosed with HIV after at least 1 CAB-LA injection with more than 30 days follow-up on ART are included. The median follow-up was 294 with a range of 35 to 422. They defined virologic suppression as HIV RNA less than 50 copies per mil, and the ART regimens were chosen locally at sites. They noted that resistance testing results prior to regimen choice were not always available, and adherence to ART, self-reported or by PK, was not available. So what this table shows is the data is divided by whether there were less than 6 months between last CAB exposure and ART start, or greater than 6 months between last CAB exposure and ART start. And the individuals were either or divided between whether they had INSTI containing ART or non-INSTI containing ART. And this was presented recently by Rafi Landowitz at CROI 2025. So what they saw was that if people had less than 6 months between last CAB and ART start, all 6 had virologic suppression, even including 1 that had an INSTI resistance-associated mutations, and 15 out of 16, or 94%, had virologic suppression, even including 7 of 16 with INSTI resistance-associated mutations. If there was a longer delay between last CAB and ART start, the rates of suppression were lower, 66% for INSTI containing ART and 60% for non-INSTI containing ART. And so finally, just one mention of lenacapavir. Looking ahead, the PURPOSE 1 and PURPOSE 2 studies showed a very high rate of success in protecting against HIV infection. there were 0 infections in Purpose 1 and only 2 HIV acquisitions in Purpose 2, which tells us that breakthrough infections, despite on-time LEN injections, are likely to be extremely rare. In the Purpose 2 HIV prevention study, only 2 individuals seroconverted on injectable lenacapavir. Both did have the N74D capsid-associated mutation. And so far, it just remains to be seen the mechanisms for PrEP breakthrough, HIV acquisition, and resistance selection with the use of injectable lenacapavir for HIV prevention is not yet known and will be an important area to continue to investigate as hopefully lenacapavir is approved soon and rolls out. But as mentioned, we would expect these breakthrough infections to be extremely rare and for lenacapavir to be a really promising agent for HIV prevention. So just to conclude, we want to emphasize that acquiring HIV on CAB-LA PrEP and other PrEP remains extremely rare. It's important as we discuss all of these HIV acquisitions and resistant to not lose sight of the fact that PrEP is highly effective for preventing HIV and should be widely used to help reduce incidence and to continue the fight against HIV. So some key knowledge gaps that still remain include what are the clinical significance of INSTI mutations selected by CAB that are detected at low frequency. We did see a number of these pop up, but so far they seem to not develop further into high-level resistance. Which CAB selected mutations have greater impact on outcomes of INSTI-based ART, and what is the long-term durability of viral suppression on INSTI and NRTIs after HIV infection on CAB-LA PrEP. And so finally, SeroPrep and other studies are addressing these questions to ultimately inform optimal ART regimen choice after HIV infection on long-acting injectable PrEP. I would like to acknowledge my MPI on the SeroPrep project, Dr. Catherine Koss, who is a wonderful collaborator and partner in all of this work. I want to acknowledge our labs at the University of Pittsburgh, both mine and Dr. John Miller's lab, who have contributed so much to this work, including many of our lab members who have unfortunately had to move on to other positions. Our colleagues at the SeroPrep and action studies, all listed here for all of their contributions to this work, and the Mosaic studies, especially Lauren Kudrick, our project manager, who was so instrumental in making all of these country teams together and the protocols successful. We have so many people in Mosaic that are not listed here by name who have generously participated in all the research studies, including HPTN. When I acknowledge them, the HPTN studies, Mosaic, Catalyst, Action, and SeroPrep, and our funders. And finally, to close, I would like to express my deepest respect and gratitude for the researchers at Harvard and our colleagues in Africa who have continued to do the work that they can despite losing so much this past spring. I hope we can stay strong together to keep HIV prevention alive. Thank you so much.
Thank you very much for your talk. And we have a few minutes left for some questions. I'd like to encourage everyone who's attending that if you have questions at this point, you can either raise your virtual hand, you can pop a question in the chat, and I will relay that question on. But we're going to start with, I think, an in-person question from Dr. Mayer.
Thanks, Patrick. Hervey, that was a wonderful talk, and I certainly hope you identify other funding sources to be able to run the bank specimens. That's such an important resource. But one question, do you have information on the test that was used? So we know that RNA testing, for the most part, is not being used in Africa. For the mosaic specimens, you have screening. But do you at least have from the sites information about whether it was a third gen or a fourth gen test? Because I think that could be really important. I think trying to get people to do RNA testing is not going to be feasible in this current climate, but the incremental difference for fourth gen may be important if you show that all these seroconversions occurred in people who only had third gen testing done.
Yeah, great question. Thank you. For all of these, it was only third gen testing. They were identified using the country's national testing algorithms, which is typically a third gen point of care rapid test. And if that's positive, that's followed by like a second or third gen point of care rapid test. So in the Catalyst study, we had collected and banked samples to address that question from all of the CAB users. We do still have them banked. They're sitting for a limited amount of time, but we have them banked from every injection that was received by a CAB user, because we thought this was the one opportunity we have to kind of look at that. So I'm not sure if we will now still be able to do that, but we do have some viral load test results from the CAB users in Catalyst. So I think it'll be interesting to see. I think for practical purposes, though, I think the third gen rapids is going to be what will need to be done for implementation.
Thank you. I have a question from the chat, which I'll try to relay here, which is, when you're thinking about comparing drug resistance across different study groups or different treatments, the question is, are there potentially confounding variables by those different study groups or treatments that might impact the results? So in other words, this might be adherence, this might be the kinds of biological risks that different groups have. And the question is, whether are those confounders or those sort of other characteristics comparable among the study groups where you're making comparisons, or is there a way to adjust for them, or is it contextual? So I hope that I'm translating a little bit, but hopefully that comes together as a good question.
Yeah, no, thank you. I think maybe what I'm understanding is, where is this resistance coming from, and can we compare that between groups? It's true that when we observe drug resistance, we can't totally be sure if it was selected because of that drug exposure in that person, or if it was transmitted from a partner, or, with PrEP, it's also hard to say with treatment starting typically immediately, what was the influence of some selection by variable adherence after starting treatment. If someone had variable adherence with PrEP, it's also possible that there's variable adherence with treatment. So I think that there could be a number of reasons why resistance is observed in these individuals. And I think with the numbers so low, right now we're really looking at these on a case-by-case basis and trying to see if we can see patterns that can help us understand the development of resistance. I think overall, though, probably what's most relevant is how these individuals do on treatment. And if treatment is successful, then it gives us some hope that even if we do see resistance with acquiring HIV and PrEP, that there's still options available, and it should not diminish or cause too much alarm over the use of PrEP.
Thank you. I have one more sort of question that I'll go back to a piece of data that I just found so striking. My mind kept wandering back. So I wonder if we could talk a little more about it. And that's the finding that in one of the studies, a third of the seroconversions have occurred in the first 3 months of starting PrEP. And the speculation was that might be due to undiagnosed HIV-1 infection in that cohort. But it seems like a lot just for window period. So what are your thoughts about how to, has that kind of finding of such a high proportion of seroconversions happening early been seen in other studies, or do you think it was something particular about this study? And what else is there anything that can be done retrospectively to unravel that? It was just a really striking finding.
Yeah, we found that to be interesting as well. I mean, it's hard to say exactly what might be going on. I think we were thinking about that when someone decides that they want PrEP, they already know that they're at risk for HIV infection. And when a new product is available, there may have been a lot of people who are trying to use that or get access to it in the beginning years. So I suspect that could have been because of our study starting right when PrEP was being introduced in the country in the initial days of introduction. So for example, we tried to start the HIV drug resistance monitoring in Kenya right around the time when Kenya was first starting to roll out PrEP. So it could be that in those initial months or years of PrEP, that a lot of people who were at substantial risk were initiating PrEP, but we didn't really, that using third generation rapids could not necessarily rule out HIV, acute HIV infection. That may over time not be as much of a issue if PrEP is routinely available and people can access it. And that's maybe one hypothesis to why we may have observed that in our study.
I also wonder whether it might be that plus, the idea of people who may have had, quite recent exposures being more motivated to come in and sign up, and it would just be in an eclipse period for diagnosis and be selecting in because of a motivating episode, that happened recently. But I haven't seen, if that's the case, you'd expect that studies would more consistently see this kind of high early, seroconversion. So I just, I found interesting.
And I think there's been a lot of work recently too, thinking about who's coming in and whether they're eligible for PrEP or if they should actually be given PEP. And so I think the non-occupational use of PEP and the expansion of that has been a really great addition to the field.
Yeah, it would sort of necessitate a different initial conversation, maybe than what's had currently in PrEP prescribing, or a more focused, history just around recent exposures. But it seems like the downside of a PEP to PrEP, introduction to antiretrovirals for prevention might make sense if this is seen in other settings. So I wanted to just give you a chance. We have about a minute left, but just your high level view is optimistic, I think. If you had to just sort of boil it down to a headline in light of all of these data, where do you see the future of PrEP in light of what's known about drug resistance? Like, what's the sort of top line takeaway, just to wrap up with?
Yeah, thank you. So I do think that it's important that we continue to have as many options as possible. I think maybe the take home is, as long as we continue using the same drugs for treatment and prevention, we will continue to have some risk of HIV drug resistance, but that risk should not be a deterrent in using PrEP and expanding PrEP, as PrEP is a really powerful, it's a really powerful agent to prevent HIV infection. So the fear of resistance should not deter anyone from using PrEP.
I think that's an amazing, concise point to end on, and I'll just echo Dr. Mayer's thanks. for your being here today, for your presentation, and for handling the questions. And thank all of our attendees. And we will be in touch with news. I think I have an announcement for our next. Ken or Alberto, if you have it, can you step in and remind us of our next session?
Mitchell Warren on August 4th at 12 noon East Coast time. So we look forward to people's participation.
We'll see you
Thank
then.
you, Irvi.
Thank you so much.
That was great.
Okay. Bye-bye.
Much appreciated.